Hello, my name is Connor O'Brien Stofa, and today I'll be talking to you about Nasophilia in Sabethes mosquitoes, blood host facial feeding behavior and mechanisms. Here you can see a picture of Dr. Robert Hancock, our resident mosquito man at MSU Denver, where we maintain colonies of 4 different tropical species. Today I want to talk to you about a very specific mosquito behavior called Nasophilia. This refers to females of a mosquito species displaying a larger than 1-to-1 or 50-50 proportion of total bites on the human nasal structure compared to the entire face. It was first observed by Dr. Woodbridge Foster with the Gorgas Memorial Lab in the 1980s. While he was collecting Sabethes chloropterus and Sabethes cyaneus species to bring back for lab colonies, they observed that in the field, to collect them, they had to put a net over their head once the mosquitoes had come down and landed on the nose. Now, this kind of facial or nasal preference has only been published in reference by 2 people, by Alexander Haddo and Pedro Galindo, in reference to Aedes simpsoni and also Sabethes chloropterus. But none of these references actually directly tested this nasophilic behavior to see specifically what was going on. Here we have a video of our first species, Sabethes cyaneus. They're these beautiful diurnal mosquitoes. They have this metallic blue and silver scaling with these mid-leg paddles, which you can see here, and they're very peculiar. They also display courtship behavior. But why we really are interested in them is because they do carry Zika virus. And here our second species, Sabethes chloropterus, also a diurnal mosquito. This time they are actually metallic green and gold scaling, and instead of having that paddle on the mid-leg, they have a white tarsi banding instead. They're also a very significant mosquito to study because they carry yellow fever. To determine whether this nasophilic behavior exists within our 2 species, we conducted a series of experiments where we collected bite count data across 8 different facial regions on our live host, Dr. Hancock. We also conducted these bite counts across 2 different treatments, one where there was normal exhalation and one where there was reduced exhalation. Here you can see a plot of the different bite areas across all the other parts of the face besides the nose for Sabethes cyaneus. And when we layer on the nose, you can see there seems to be a significant biting preference in the nasal region. And for a little frame of reference, here is an example of some nasophilic behavior we observed of cyaneus during one of our live trials. As you can see, they vie for spots on the very tip of the nose. On that bioassay chamber you just saw Dr. Hancock in, it is an open air structure, and we draped a mosquito net over top of it, and each of the 20 individuals per trial that we included were first screened for appetitive blood-seeking behavior before individually being distributed into the chamber for testing. Back to our bite distributions. Here we see an example of chloropterus and its bite distribution across the entire face. And then once again, if we layer on the biting distribution on the nose, it also seems to have a nasophilic behavior just like Sabethes cyaneus. And here we have an example of that nose biting behavior by chloropterus in the lab. So our big first question was, do Sabethes species display nasophilic bite preference? To determine that, we looked at chi-square goodness of fit test for both species, and we found yes, they do, but only the Sabethes cyaneus species. They have a statistically significant bite preference for the nose, and as you can see here, chloropterus had much closer to a 50-50 distribution of biting on the nose versus everywhere else. And so, no, it was not a significant nasophilic preference. To dig a little deeper, our second question asked whether reduced exhalation affected Sabethes cyaneus bite efficacy. When I say normal exhalation, I mean unlabored nasal breathing with the mouth closed. Reduced exhalation was a little bit tricky. We ended up stuffing the nasal passages with disposable wipes and having Dr. Hancock breathe through a vinyl tube that exited outside of the chamber, as you can see by the red arrows there. Back to our second question. Reduced exhalation was found to have a significant difference over regular exhalation when it came to cyaneus. You can see by the red coloration on the frequency map here that when there was exhalation, we had a very strong nasophilic behavioral signal, but when we reduced exhalation, it was almost completely removed. And in our final question, we asked whether reduced exhalation affects our other species, Sabethes chloropterus, in its biting efficacy. And through another chi-square of independence test, we found, yes, there was a significant difference between the overall bite efficacy between exhalation and reduced exhalation, as you can see here in the colored chart. So a quick summary of our initial findings. When it comes to our live host testing, in laboratory conditions, we did find nasophilic behavior significantly in Sabethes cyaneus, but not in Sabethes chloropterus. At the same time, we did see that normal exhalation, normal breathing, did seem to be a larger gating tool for them to find a blood host, and we did see a significant decrease in bite efficacy in both species when it was reduced. So now that we've found that there is a nasophilic behavior happening here, we need to tease apart the actual stimuli that are involved in driving this behavior. And the odor plume, which is, you can see here, is a really good example of showing all the different variables, the concordant sensory cues that are involved in mosquito host-seeking behavior. Through positive anemotaxis, the mosquito moves through an odor plume and it encounters CO2, it encounters humidity, heat, and finally visual cues in that order as it gets closer and closer to a host. So we wanted to create a model that could tease out those specific stimuli, and we call him Hot Bob, and I'll explain why in a minute. And it will allow us to separate out factors like CO2, humidity, heat, and even the location of air passages. To really simulate a real host and a real odor plume, we needed an experimental model that could simulate normal skin temperature, which we did with a heating pad applied to the inside surface of the face, and also normal breathing. So we also have these tubes that go outside of the bioassay chamber where an observer can breathe into them, and they can have normal exhalation coming out of the nose, the eyes, the ears, wherever we want to place those tubes. We did find some interesting data just conducting our controls on the Hot Bob model, as you can see here in a little video. Just with visual and heat stimuli, we got Sabethes cyaneus to display that same nose clustering that we saw with the live host, which is really exciting. We've seen here that the nose is a really interesting structure. There's a lot of reasons why we might be seeing this nasophilic behavior. For one, as a source of blood and sensory variation, it is unique because it is the coldest structure on the face. The nose is made out of cartilage, and it doesn't have direct subcutaneous blood flow. It has smaller capillaries, and it also doesn't have as many nerve endings. For all of these reasons, there's a lot we can still dig into why this nasophilic behavior is happening. In conclusion, we have discovered today that nasophilic behavior does indeed exist within the Sabethes cyaneus species. We don't know why, and there's still a lot of really interesting concordant cues to tease out to figure out. Monkeys don't have hair on their face the same way that humans don't. And for that reason, there may be some similarities in why cyaneus would be all biting humans in addition to their normal sylvatic species for that vector cycle. And this is extremely important. We need to understand this behavior because as the forest and urban gradients in Panama and other tropical places in South America and Central South America get smaller and smaller, these disease vector cycles will cross over. So the more we know about this nasophilic behavior, the better we can be prepared for the future. Thank you for your time, and I hope you enjoyed my presentation.